We are going to attempt this item, respond to it, and score. Fish farmers in Kalangala district observed reduced movement and slow growth in tilapia after cassava processing wastes were released into nearby cage waters. The fish were also fed on preserved commercial feeds. Table: physiological indicators in tilapia. Pause and enlace the date table. All the parameters in the affected fish decreased. What may be the cause? Further findings showed feed chemicals attached to digestive enzymes and water samples contained cyanide compounds from cassava wastes. Task Part A: analyze how enzyme inhibition and cyanide poisoning affected digestion, ATP production, and muscle activity in the fish. Part B: evaluate sustainable practices that could restore fish health and improve productivity. Responses: we are going to analyze. Format: start with the cause, what happened, why, how, mechanism/process, and outcome to get full highest score. A P, which is 3 scores. The preservatives in the feeds acted as enzyme inhibitors by binding to the active sites of digestive enzymes, reducing the formation of the enzyme-substrate complex. This lowered the rate of hydrolysis of food molecules into absorbable nutrients such as glucose, fatty acids, and amino acids. Reduced absorption of respiratory substrates decreased the supply of glucose to muscle cells for cellular respiration, lowering ATP production required for muscle contraction and active swimming, thereby reducing movement and slowing growth in the fish. Before we continue, this lesson is sponsored by ABE, the Association of Biology Educators. They have prepared the following students' biology national seminars. Note the date and the contact phone number. Another analysis response: cyanide compounds from cassava wastes inhibited cytochrome CE oxidase in the inner membrane of the mitochondrion during oxidative phosphorylation. Electrons failed to pass along the electron transport chain, preventing pumping of hydrogen ions and reducing activity of ATP synthase. As a result, little ATP was produced despite oxygen uptake, leading to muscle weakness, reduced swimming speed, and poor survival of the fish. Task Part B: evaluation of strategies. Proper treatment of cassava wastes is effective because it removes or reduces cyanide compounds before release into water, preventing inhibition of cytochrome CE oxidase and allowing normal functioning of the electron transport chain and oxidative phosphorylation for adequate ATP production, required for muscle activity and survival. Using safer feed preservatives reducing harmful preservatives prevents blockage of digestive enzymes, allowing normal formation of the enzyme-substrate complex and efficient breakdown of food into absorbable respiratory substrates such as glucose, which supports normal cellular respiration, growth, and movement in the fish. Thank you for watching. Subscribe for more updates.